I'm Kelly Loyet. I'm a senior scientist at Genentech. I work in research at South San Francisco in California. And so I work in the biochemical and cellular pharmacology department. I work in the biochemical and cellular pharmacology department, and pharmacology is the study of how a medicine affects a biological system, as well as how a biological system affects a medicine. And so we're more interested in the latter, and the medicine we are looking at was lampalizumab, or anti-factor D, which was in clinical trials for geographic atrophy. What we were interested in was looking at the stability and the efficacy of it in terms of looking at it before it actually goes into patients. We had made some different versions of lampalizumab in order to allow it to be dosed at higher amounts, and thus it would be in the eye for a longer period of time. We were wondering if, when it's in the eye for a longer period of time, would it remain stable and would it remain active? So for this study, we looked at first how lampalizumab, as well as the different versions of lampalizumab, would it actually still bind to factor D after 4 months? And so we were able to obtain some surgically removed vitreous, and looking periodically through the 4 months, we looked at the ability of it to bind, as well as potential chemical reactions that it could occur that would cause it to not bind to factor D. What we found was that it was very stable after 4 months, and there were very few chemical modifications, particularly deamidation is what we looked at. And so we then looked in a preclinical model, and we looked in this preclinical study how it would be stable after a month. And so that was a little bit more relevant than a in vitro or a study that was just done on the bench. And in this study, it was also remaining active. It was remaining stable. And from there, even though a preclinical study may tell you that a molecule looks great, it may not actually translate to what you would see in patients. So we're very fortunate to be able to obtain some aqueous humor samples from the Mahalo phase 2 clinical trial. So these aqueous fluid samples were from patients dosed with lampalizumab, and they were dosed either once a month or once every 2 months. So basically it was very stable after 2 months and after 1 month, which would indicate the stability of it. So in a nutshell, we were able to see that we could translate our studies in the lab to patients that were actually dosed with the molecule, and it enables us to better predict whether our molecules will remain active and will remain stable over long periods of time in the eye. So lampalizumab did not meet its primary endpoint in the phase 3 clinical trials, which is very unfortunate and disappointing to everyone, although there is a lot of work that's going on in terms of analyzing the data and analyzing samples from this study in order to try to figure out why it didn't work. Genentech is very committed to developing therapies for geographic atrophy, and there are other molecules that we can apply this same strategy in order to better predict whether or not a molecule will remain stable over long periods of time. And it's very important that we develop longer-acting delivery techniques so that patients have fewer injections, there is less of a burden on the patients, more compliance, and potentially better outcomes.